Hello. Hello.
Yes. Yes, sir.
Dr. Tanzil Ahmed, can you hear me, please?
Yes, sir, you are audible.
Oh, thank you very much.
Sir, audible, sir.
Yeah. I can hear you. Hello, Dr. Tanzil. Dr. Tanzil, can you see me? Can you watch me?
Yeah. I'm able to see
Okay,
you.
so yeah, so I need a little guidance how to connect my presentation, how to show my PowerPoint, because I have not used this on Google. I'm used to Zoom. Ask Tanisha if she knows. No, it's okay. Hello. So now I'm logged in with my Google. But do you know how to connect my PowerPoint presentation? Okay, wait for a while. Wait for a while. Yeah, I'm trying to connect my presentation. Please wait. Huh? Okay, wait. So I'm just doing some setup. I will be back with you guys. From here. Where is that? Oh, that's what I was doing. Wait. No. Wait. This is this one. So I have to go back here. Yeah, and then yeah, present now. Huh? So then your entire screen. Huh? Shall I go on this one? And then I think from there I have to open this one. Huh? Okay. This one. No. So then I have to go. And then how to go back? No. And where is I have to switch on my speaker, everything. Right? Speaker is on. Okay.
So I think the
is visible. Right now, screen is visible and we should start.
Okay, thank
Tanjeel
you very
sir,
much. Thank you very much.
should we proceed? Tanjeel Ahmed sir, should we proceed? Tanjeel sir. Hello. Okay, so I'm starting. Honorable Chancellor Sri Rajiv Kothiyal sir, respected Pro Chancellor Sri Abhinav Kothiyal sir, Honorable Vice Chancellor Professor Mahendra Pandey sir, Registrar Sri Sanjeev Agarwal sir, Pro Vice Chancellor Professor Rahul Kumar Misra sir, all the directors, our keynote speaker Dr. Muhammad Khalid Parvez sir, all the faculty members, and my dear colleagues and dear participants. It's a matter of pleasure that we all are connected on this virtual platform to learn and discuss about the COVID-19 virology, pathogenesis, and containment. On behalf of entire IFTM University family, myself, Professor Arun Kumar Misra, welcomes you all in the today's webinar. We are fortunate enough to have Dr. Muhammad Khalid Parvez with us to deliberate his views and his think tank on COVID-19, virology, pathogenesis, and containment. Before starting and handing over the mic to our keynote speaker, I would like to share his brief profile. Dr. Khalid Parvez is currently working as professor and DSR principal investigator in King Saud University, Riyadh, Saudi Arabia. He has done his UG Bioscience from Jamia Millia Islamia, New Delhi, and MS in Biotechnology from University of Calicut, PhD in Virology from Institute Center for Genetic Engineering and Biotechnology, Jamia Millia Islamia, Central University, New Delhi. His research interest is in the area of hepatitis, virology, liver diseases, recombinant baculovirus, insect mammalian cell expression system, and computational drug designing and discovery. Professor Parvez has worked as doctoral and postdoctoral fellow from ICGEB New Delhi on hepatitis and molecular biology. Additionally, he has served as Forgatory International Visiting. He is serving as reviewer and editorial board member in number of research papers and research journals. He has to his credit approximately 20 national and international awards, 86 research papers in various journals of high repute and in good impact factor. Now I request Professor Muhammad Khalid Parvez to share his think tank on COVID-19, virology, pathogenesis and containment. Please, sir.
Hello, everybody. Namaskar and salam to everybody. This is Dr. Khalid Parvez, and it's my great pleasure to be with you guys, and especially reconnected with my all Indian academic fraternity. And a special thanks to Dr. Tanzeel Ahmed, who actually initiated this process, and I gladly accepted his request. Despite I am not currently in Riyadh, I am traveling to see a family to other city, but I could manage that one. And I think without any delay, I should rightly start with my presentation. And before I start, I should make that, of course, I have my training and education in virology, but COVID-19, as everybody, this is also very new to me, and I have also initiated some work on this virus, and we have published a few articles on that also. So let us start with my presentation. Can you see my slides? Can you see my slides?
Yes, yes, we are, it is.
Okay, thank you. Okay, so let us just start the introduction with a very an old Hollywood movie that was named Contagion, and I hope many of you guys have seen this movie and have experienced virtually what happens in such pandemic situation during any viral or bacterial or any such kind of infection disease outbreak. So I'm not going through the details of this movie, because that time we only imagined, we only experienced the scenario, what people can feel and how the society reacts with this kind of situation. But unfortunately, we had to face this one in reality. And since the past almost 10 months, we are facing this global crisis. Okay, so because it has been almost 11 or 12 months of announcement and the pandemic of this disease, many of us are very much aware and have a good knowledge about this virus and the disease. So I will not go in very minute details of the virus, but of course, this is an academic platform, and we have undergrads students who are not from virology and who are participating in this meeting. So of course, I have to give a little bit introduction to those group of audience. Okay, so coronaviruses have derived their name because of their shape, the crown-like their structure, and they belong to the virus family Coronaviridae, and they have 4 genera: alpha, beta, gamma, and delta, depending on their genetic sequences and their relation to each other. And of these, beta coronavirus is the most infectious human coronavirus, and it has a spherical enveloped virus with a very large RNA genome, about 30 kb, and it has a very potential zoonosis in mammals, and especially bats, the civet cats. The civet cats are the main native of China and the neighboring regions, and pangolins. And they generally cause respiratory tract infection and manifest as common cold, and it's mild to severe pneumonia. If you look back to the previous human coronavirus outbreaks, we have OC43, HK, that is, stands for Hong Kong, U1, 229E, and NL63, and they are the most common circulating virus strains of human coronavirus, and they cause common cold, and you resolve in 10 days or 1 week or maybe 2 weeks, and you don't need hospitalization and special medication. Okay, so after that, we experienced another coronavirus that's called SARS coronavirus, that stands for Severe Acute Respiratory Syndrome virus, and in 2002 and 3 in China. And then in the Middle East, especially in Saudi Arabia, people experienced another emergence of another coronavirus that's called MERS coronavirus, that is the Middle East Respiratory Syndrome virus. But these 2 recent coronavirus, SARS and MERS, were very soon contained in means of that it was controlled based on the all government health authorities and the medical authorities, WHO involvement. So it didn't cause much of devastation as we are facing nowadays. So when I talked about the reservoir, potential reservoir, the bats are the main culprits, main reservoir of this virus. Not only this virus, but also it has been known for other viruses that infect humans and cause disease. That's called, if you see in the this one yellow highlights, Marburg virus, and then Nipah virus that has also re-emerged recently in Southeast Asia, and then Hendra virus, and of course what we are talking about, the SARS coronavirus, and other encephalitis causing viruses. So now we should look into again revisit the genesis, how this virus emerged and how did people got infected and what is the actual origin. But still we don't know officially that the actual point or place of origin, and how this virus started, and what was the first animal from where we got the virus, and who was the first contact person who spread the virus in China and outside. So of course this virus is novel, very new, and it was first announced in Wuhan, China. in a wet wildlife market. It is expected that it started from that market, from the infected bat or civet or pangolin. And this is the 7th human coronavirus reported now, and the 3rd most pathogenic after SARS coronavirus, that is called SARS coronavirus 1, in relation to this new SARS coronavirus 2, and MERS coronavirus. And if you look into the epidemiology and the figures, as per yesterday, that December 3, we have this much global positive cases reported and about this much of deaths, including our own cases in India. So the outline is that the bat, especially called the horseshoe bats, they are the primary and the main source of the virus, and somehow it spilled in the market where people eat the bats and related mammals, especially in China. And from there, it jumped into other mammals like civet cat or pangolin. And from there, most likely, it went to the eaters of these wild and weird animals, especially in China. Okay, so SARS coronavirus is based on its genome sequencing and phylogenetic analysis. It was seen that about 96% similarity with bad coronavirus. And the strains are this one, ZXC21, ZC45, or especially the RATG 13. 13 means 2019, year 2013. But it has less, genetic severity, about 80% with SARS coronavirus 1 that emerged in 2002 and 3 in China. But genetic, sequencing and genetic information connected this new virus to bat viruses, especially the SARS, bat-like SARS coronavirus, that is abbreviated as bat SARS-like coronavirus, and the pangolins virus, about 91% similarity. And also, it is there is theories that it also evolved naturally, and it jumped from one species to another species, and then it came into the human, where it evolved, and then it adapted, and then it started causing disease. That is called the manifestation of this new virus. Apart from this, there are other theories circulating that it was lab-made, it was developed by some people, maybe from China's lab, as a bio weapon or something, but it is not officially confirmed by any world authority agency. So officially we cannot say like that. Okay. So in terms of nomenclature, provisionally it was called 2019 novel coronavirus, and WHO officially named it SARS coronavirus 2. And its main etiology and disease is the same I told you: severe acute respiratory syndrome related to your lungs infection and pneumonia. And then we have to look into the next slide for its mode of transmission and how it circulates among the society or from people to people. So the direct human contact has been established not only for this SARS coronavirus, but for other coronaviruses, that from sneezing, coughing, and the spread of nasal droplets, oral mucosa when we talk and when we laugh or when we speak, and it goes in the closed environment, and through the aerosols it infects the other healthy persons, and its half-life is about 3 hours. And also it has been not in clinical samples, but in laboratory settings. It has been shown that it can survive on copper surface for 4 hours, cardboard for 24 hours, steel and plastic surfaces for up to 72 hours. So that's why it's always recommended to sanitize and keep your all surfaces, hand, body, clothes, and every household things, and every door, table, whatever you are using, to sanitize regularly and allow the virus to be inactivated or killed. And what are the other potential routes apart from your nasal or oral mucosa droplets? The gastrointestinal sheddings, that patients who are corona positive, if they shed, if they defecate, the virus comes in the fecus, in the urine, and when it's contaminate the water, as for the other water borne diseases like hepatitis E or A or like diarrhea or something, it contaminates the water. And if you don't have well regulated water bodies, well cleaning or sanitization or water purification, etc., sewage management, it comes in your drinking water sources, and then if you ingest it infects you. Like, so that's why it's called fecal oral transmission. And if you just compare the incubation period means the day the virus infected your organs, your body, and it started replicating itself. And after replication, it starts the onset of the disease, and your symptoms starts coming out. So for SARS coronavirus 2, what we are talking about, the COVID, actually, interchangeably we can use COVID virus, SARS coronavirus 2, COVID disease, corona disease. So sometimes we change the wordings. And it is 2 to 14 days. For seasonal flu, I talked about your normal coronaviruses, 2 days, and the coronavirus 1 for 2 to 7 days, and for MERS, 2 to 14 days. So it's very close to the MERS coronavirus in terms of incubation. But if you relate its genome sequence, and if you compare the two viruses genetically, it is far from MERS and SARS both. And again, I'm reminding you, it's very close to the bat and pangolin coronaviruses. So it's very strange with this virus. If you compare with the other related viruses, it is not affecting your lungs only. It is affecting almost each and every part, each and every cell, or each and every organ of your body. So right, so about 80% of cases are asymptomatic. That means you have the virus, but it is not manifested. You are not having any sign and symptoms that you have acquired this virus. Just sometimes you have mild fever, cold, headache. Out of this, about 50% cases are symptomatic. That means in 50% of infected population, the virus is manifesting. It's causing the disease. And what are the types of diseases? Mild pneumonia, high fever, breathlessness, you need hospitalization. And out of this group of symptomatic patients, about 5% cases are affected, and they develop severe pneumonia. And after that, you have lung failures, ARDS, septic shock, and death. In the normal population, old age people are more susceptible, more vulnerable, and especially those who have comorbidity, that patient who have other chronic diseases like heart disease, kidney disease, liver disease, that pre-existing before you catch the virus, or diabetes. They are the most vulnerable people who get the most severe kind of symptoms and prone to death. And what are the uncommon? But now it's getting more common. People who are affected with coronavirus and having severe disease, they develop COVID toes, that is called rashes on toes and fingers. You have red rashes on your finger, in your feet. Skin decoloration, burning sensation in your body, dizzying, fuzziness, like you are sometimes feel lost, just like you are drunken, or you have some psychological issues, and also loss of taste and smell. So these are the uncommon manifestations. But recently I see some reports that loss of taste or smell is not only the disease symptoms, but this is the sign of recovery. People who are in the recovery phase, for those, taste and smell are good signs. If they are losing taste and smell, it is said that in clinical studies they are now recovering and they will be like healthy very soon. Okay, so just have a look into how this disease is developing, and of course your body immune system is playing an important role in this one. So not very in-depth and very good data is currently available in regard to COVID-19 or SARS coronavirus 2 regarding the immune modulations, but we have little bit informations now that the first thing is this virus is very new, and we had no exposure previously. So the population has no pre-existing, that's called herd immunity or trend immunity against this virus, and the naive populations have delayed adaptive immune in the body. Unlike coronavirus 1, most spread of coronavirus 2 is done by asymptomatic cases, and the cytokine and other inflammatory molecules are playing very important role in one. So one is called IL-6, interleukin-6, that has been seen previously in the case of SARS and MERS coronaviruses, and that's why it is speculated and expected that the same molecules are also playing in establishing pathogenesis in COVID-19 patients. And then you have the IL-6 receptors, and that has been you have another blocking molecules, the antagonistic molecules, that is called sarilumab or tocilizumab. They are the known antagonists, and the same molecules are right now in clinical phase 3 trials that are very important molecules that block the IL-6 molecules, and that's why it's called, like, immunomodulatory drugs against coronaviruses. So now I should move to next slide. So what we have, like, different tissues and organs that are infected by the virus, and we have different kinds of symptoms related to almost all organs and systems in the body. So first we see the gastrointestinal symptoms. What we have is vomiting, stomach pain, diarrhea, and virus is shedding in your gastrointestinal tract specimen, stool, or also in some urine. Hepatobiliary or pancreatic infections are also, we have this virus symptoms and affecting it's the manifestations in pancreas, bile ducts, liver, also in your cardiac system, in your heart or other myocardic tissues, and also some neurological signs and symptoms that's called Guillain-Barré syndrome, that is affecting your immune system, immune-mediated nerve cell damage, encephalitis, and stroke in COVID-19 infected patients. Now we should look into a little bit biology of the virus. And we see that the electron microscope figures are available. And this is the real photograph, EM photograph of the virus. And this is the cartoon figure, and we see the structural details of the virus. The outer, spikes are called the surface protein of the virus, S protein. And then we have, next is membrane protein. And then we have nucleoprotein that actually encloses, and it keeps your the viral RNA or DNA intact, protected from the cellular or outer environment pH and whatever is that degradation from those, and it protects. And then you have envelope protein that's called the E protein. And interesting, we have in other related coronavirus, that is the coronavirus 1 or MERS coronavirus and others, that is called the hemagglutinin stress protein, that's called HE protein of the coronavirus, and that is absent in this virus, coronavirus 2, because of the mutations. And all these mutations have very good implications in its mode of like high transmission, high infection, high pathogenesis, all these things. Okay, so how this virus is getting into the body? So we have a very known cell receptor that's called angiotensin converting enzyme 2, and that is expressed in almost all the cells, organs, tissues of human body in a very high amount. And this is also expressed in bat, civet, pangolin, apart from humans. And that's why it is like zoonotic, and because it has the same receptor in bat or civet or pangolin, like mammals, and when the virus is coming in human body, it recognizes the same kind of receptor in humans, and they are getting attached, and then by the cellular uptake mechanism, it gets internalized inside the cell, and when it is inside the cell, the viral biology works, and with the help of cellular mechanism, the virus uncoats, the RNA is released, it gets, it expresses all its proteins, then virus encapsulation, viral, morphogenesis in nascent viruses, they exit the cell, then go out and circulate in your system, infect the new naive cells. And that's why it spreads all over the body. Then it causes, in connection with host systems, host immune systems, host other mechanisms, the disease onset, right? And also, recently there is another receptor that's called dipeptidyl peptidase that has been shown in MERS coronavirus. Also, this virus uses, apart from S2, this DPP4 receptor is also used by SARS coronavirus. But officially, experimentally, it has not reconfirmed. And if you see at the Genome Organization, we have the genetic structure organization of this virus. The first one is from 5' to 3' and the ORF1 1A, ORF2 1B. Then these two are called non-structural proteins. They help the virus to replicate in others, like replication process, enzymatic cleavage, processing of the virus, that's called protease, all these functions. And the structural protein, which actually make the virus viral structure, that's called the surface, envelope, membrane, and nucleocapsid proteins. So what is the highlight of this slide is the non-structural protein that's called 3A or 3B, right? And then you see the different non-structural proteins called 6, 7A, 8, 9B. These are called the accessories protein, right? So we have all in the operating frame, total 12, 2 of your non-structural proteins, 4 of your structural, and 6 is again the accessory proteins. So the total non-structural proteins expressed by the 2 non-structural operating frames are 16 in general, and a structural protein you have total 5. But in as I told you in previous slide, in this virus, COVID-2 novel virus, the hemagglutinin stress is not present. So if you compare with other coronavirus, it has 4 proteins instead of 5. And accessory proteins in coronaviruses we have 6, because one is missing in this virus, COVID-19 2 virus, coronavirus 2 virus, but it is present in MERS and coronavirus 1. So we will talk later on about this one. So this is an overview of how many total proteins and non-structural proteins it is expressing, not only this coronavirus 2, but coronavirus 1 and MERS, and what are the other functions. So so for example, the important non-structural proteins I will just highlight here, that's called the NSP12, that is called RDRP, that's RNA-dependent RNA polymerase. That has been targeted using the in silico approaches, and also later on by using the related viruses, RNA replicase, RNA polymerase inhibitors. That's called a kind of remdesivir, for example, or falcipaviir. They are the RNA inhibitors, and those are well working for this virus. And for example, there this one, another virus is called MPRO3CL protease. That protease, and for like hepatitis C virus or HIV virus and other RNA viruses, there are many drugs in the market that targets the protease of the virus, and then they actually stop the virus replication. So this NS5 non-structural protein or 3CL protease has been also researched for to develop the antiviral agents. Now we see the accessory proteins. I talked to you about that. So accessory proteins are basically participating in modulating the host immune system, that is of course in favor of the virus, to establishing its replication and causing the disease by down regulating host factors, including importantly the immune system. And The 3B, a non-accessory protein, as I told you, it is absent in this virus. And what is the function in other coronavirus? The 3B modulates the type 1 interferon production and apoptosis and induces the cell cycle arrest. And you know, the interferon is the acts as antiviral molecules in the body, and they cause they help you to fight against the virus. And because in COVID-19 2 virus, the 3B is absent, so maybe not proven experimentally, they are also weakening or downregulating your interferon production and your immune system to fight this virus. So now we will see a little bit comparative study assessment of this virus and genomic comparison of SARS coronavirus 2 with the previously known SARS coronavirus 1 and the MERS coronavirus. And this is the little overview of genomic structure. We have 4 in MERS, but we don't have in coronavirus 2 and coronavirus 1, like this. And here, like, you know, we have 6 and 7A, but we don't have 8A. We have only 8. And here in SARS coronavirus 2.1, we have 8A and 8B. Okay. So we have also done some our own studies. I have my old friend who is working in US, and we have done a study, and the paper is right now in press. So we have done some genome-wide, comparison. That's called multi-genomic sequence comparison, and not picking the all SARS coronavirus, but we picked the most important and widely circulating viral sequences. Two for the coronavirus 2. One isolated from Wuhan, China, and one from USA. And SARS coronavirus 1, 3 strands, then one cold causing virus, that's OC43, MERS, and also from the other mammals, as I told you, the civets, the bat, and pangolin. So based on these studies, we concluded some of our results that what we found is the SARS coronavirus 1 has only 79 or 80% similarity with SARS coronavirus 2 COVID, and it has about 99%. SARS coronavirus has 99% similarity with civet, and SARS coronavirus 2 has 79% similarity. But it's very important to see this one, this bat coronavirus, that the 2013 isolate from China. The SARS coronavirus 1 has about 94% genomic sequence similarity with bat, and our COVID-19 2 virus has 96% of genome sequences. And these are the protein sequence similarities or surface protein similarities of this virus. Okay, so now we should look into the spike protein of SARS coronavirus 2, because this is the protein which binds to the cell surface receptor that is that we knew, that is called ACE2, the angiostensin receptor binding receptor. So the surface protein or of the virus has a RBD, that's called receptor binding domain, and that is the major antigenic site. And I should also tell you right now, this is the region used for the vaccine development. If you are making protein-based vaccine, that's called sub-unit vaccines, this is the protein part of the virus sequence that is being used to produce vaccines, protein-based vaccine, I mean. Okay, so it has 6 amino acids, important antigenic amino acids, that's in the SARS coronavirus RBD, and 6 out of 6, 5 differ. 5 differ means we have the difference of 5 amino acids mutations in comparison to SARS coronavirus 1 or MERS coronavirus or other bad coronaviruses. So we have 5 amino acids mutations in coronavirus 2, and because of these mutations, it is interpreted, it is highly recommended or suggested the binding, strong binding of this virus surface, protein is with the human receptor, and if you have high binding capacity, the virus is very strongly attached, and they are infecting your body. There is another very important region in the surface protein that's called unique furin cleavage site, that is 12 nucleosides insertion mutation in the virus. If you see in the structure, that is the junction of the surface S protein 1 and S protein 2. That is left hand side we have the C terminal, N terminal, and right hand side we have the C terminal of the protein. And here at the junction, this virus, this SARS coronavirus, has acquired insertion mutations of the PRRA, that is the amino acids, and that generates a special unique site in the protein that is called furin cleavage site. And what is furin cleavage site in summary? That actually makes your virus protein forced to change its conformation. And you see in biology, the protein conformation is very important for its activity, and when it is going to interact with some other protein. So the conformational change is very important for any protein for its functionality. So this introduction of this furin-like site is causing, is speculated, and it has been suggested to play another important role in the binding of this spike to the S2 receptor. And it has been already shown that this kind of cleavage, furin cleavage site, is plays a very important role in some other RNA related RNA human viruses before. Okay, so now we see the other mutation, the deletions in conserved 3A aggregation motif that I told you, the accessory protein, the absence of 3B, and this is conserved among all beta viruses, and they regulate surface expression, interaction, internalization of these two proteins. And there is an absence of diacetic, that's called EXD, the 3 amino acids moiety in the crucial internalization motif of this virus. And the 3B is absent in COVID-19 coronavirus 2 because of generation or introduction of 6 stop codons. Naturally, what the isolates we have as right now in public domain, so we don't have this ORF3B protein because of introduction of 6 stop codons. And finally, only 6 instead of 7 accessory protein we have in SARS coronavirus 2. Now another information is coming from the genomic research. The another important mutations in coronavirus 2 genome is the papain-like protease region, this one. So cysteine, 14 amino acids insertion in this region. You see the green color. So what is the cysteine protease? So cysteine protease is a kind of protease in proteins, especially in RNA viruses. And RNA viruses, they express a very large polyprotein. So this polyprotein is a multi-domain protein, have different functions of the different small proteins, and the protease actually cleave the long polyprotein into a small different proteins, and then they become functional. One is like doing replication, one is doing some guidance activity, one is DNTP binding activities, one is or interacting with other host factors. So there is another insertion of replicase that is called P1AB insertion in SARS coronavirus genome. So we have seen many unique mutations, especially in SARS coronavirus 2 isolates. Now we should look into what we talked about, the control measures, including treatment, intervention, interventions, the drugs, antiviral drugs, the vaccines, the making containment zones, precautions like sanitization, mask, distancing, whatever. Hello, can you are you guys hearing me? Are we connected? Are we connected?
Yes, yes.
Yes, sir.
Okay. Thank you. Because we were not talking, so I just made it short. Okay, so now see what drugs we have. Actually, nowadays nobody is talking about drugs. This is very interesting because now some vaccines are coming in the market. We will talk in the next slide. So everyone is talking about vaccine. And previously, before 3-4 months, when we were in the lockdown period, like drugs were very important topics of discussions. So anyway, so now what drugs we have now is the important one is remdesivir. So actually, it is the repurposed drug. That is the drug used or developed for other related viruses. I think for the Ebola, and now people tested it for this new virus, and it's working fine. So now it has been in phase 3 clinical trials and already approved in some other countries for emergency use, especially US or Russia, Japan, or some other countries. And other one was the first one that came into limelight was this hydroxychloroquine. And later on, there was a controversy, and trials of this drug was stopped, and now it is not very much recommended. There is another drug called leponavir or ritonavir, that is the RNA replication inhibitors. And other, I talked to you about the immune modulators, the immune targeting drugs, that is called the Nkaina or canakinumab or tocilizumab or siloximab. These are the inhibitors of IL-6 molecules, and that is the immune system modulators, immune modulating drugs. That is also working well for this coronavirus too. And these four are now in phase 3 clinical trials. And in emergency cases, also in hospitalized and severe cases, these drugs are now being given to the patients. So now I'm moving to the very interesting and most talked area of treatment. It's called the vaccine, that is the prevention mode. Okay, so most likely this is my last slide. So let us discuss what vaccines we have. So for the undergrad students and people who are not actually from the biology and who are the audience present with us, what are the vaccines? Are the preventive mode of treatment. You get the antibodies basically against the antigen, and you inject in the patient. So if you are injecting the ready-made or available antibody to a very specific particular virus or bacteria, that is called passive immunization, right? And that is also a kind of vaccine, but not in the real sense. And what are the real vaccines? So real vaccines are actually the product of the virus, which is immunogenic, which is antigenic, that your body recognizes very well, very easily. So you inject in the body that part of the virus, and it mimics the virus, and your body or your immune system recognizes this as a real virus, and the immune system comes forward and attack this protein, and they make their own defense system. They develop and produce the antibodies, actually the barriers, the security person, the soldiers to fight the virus. So currently, if you see the COVID-19, so we have about 41, as of yesterday's data, the current data, we have 41 vaccine candidates in phase 1 trial. We have 17 vaccine candidates in phase 2 trials. We have 13 vaccine candidates that has almost completed the phase 3 and final trials, and some of are in the production phase. I will talk later in down. And 7 vaccines are almost getting approval for some limited use in some special circumstances, not in all cases, with a special permission of the government. But so far, none of these have been officially approved by FDA US or India or WHO or UK, any government health regulatory or drug regulatory authorities. Okay, so now for the especially for the students, I have made have a breakup of the a type of categories of vaccines. So let us first talk about the genetic vaccines. What are genetic vaccines? So if we 12th standard, if you go back to our 12th, 11th, 12th standard, or maybe nowadays 10th standard biology book, we have that's called the uh, central dogma of genetics. What is the central dogma of genetics? We have our genetic material, your DNA, right? Now the DNA is transcribed into mRNA, RNA actually, and then the RNA is the main molecule that produces your proteins, and then the proteins undergoes different changes, and it has its own functions. So from DNA you have RNA, then you have protein. So now the vaccines are made at all three steps. So the old category and old fashion was to use the proteins, the third step. Now we have gone with the advancement of our technology and biology research, biological knowledge on genetics, biotechnology, everything. So now we have gone far ahead, and you have used the reverse genetics basically. So we have now viral vector based vaccines. So what are the genetic material based vaccines? So now the front runner of the COVID-19 vaccines, we have now two vaccines coming first compared to others, are mRNA, that's called messenger RNA based vaccines. That has completed phase 3 trial, and also the that's called Moderna. That is Moderna is a company, and then AstraZeneca Oxford vaccine, and another one is Pfizer. So the Pfizer vaccine and Moderna vaccines are mRNA. So what we have is that instead of translating the protein, purifying the protein, and injecting the protein in the body, we have now direct mRNA. And RNA viruses have very unique replication properties. If they have RNA, that is the direct coding, that is called the plus strand viruses, and the sense, that is they directly code the protein. They don't need to go into complementary RNA. So we have in the injection, we have the vaccine mRNA, and when the mRNA is injected in the body, it's going inside the cell or other mechanism, and then they are directly coding the protein of the virus surface. And when the protein is produced inside the body, it is antigenic, it is immunogenic. Our body system is, reflecting. They are fighting, and they are producing high amount of antibodies, and then it is protecting the body against the virus. The same thing is with the Pfizer, vaccine. The third one is AstraZeneca and Oxford UK vaccines. So instead of mRNA, they are using the viral vector, that is the adenovirus based vaccine. And what is this strategy? You put your genomic sequences, your DNA of your protein, whatever virus, bacteria you want to express, in some other known viral vector that is not causing any harm in your body, but they are very fast replicating. And fast replication means they are producing the antigen, the vaccine product candidate, very high amount in the body, very fast in the body. So these are adenovirus best vaccine. Another vaccine is called Sputnik 5, that has been already produced and already been approved in Russia. And now we have completed Johnson and Johnson company, the gene-based vaccine. So we have 5 gene-based vaccine: the Moderna, Pfizer, the Russian, Sputnik, AstraZeneca, Oxford, and Johnson. So all these are having already done phase 3. Now I told you the viral vector like adenovirus or other viral vaccine that's in phase 3 trial right now, Novavax or Medicago, GlaxoSmithKline vaccine. Now you see protein-based vaccines. So what we have done is in a reactor or in a bioreactor, you produce large amount of antigenic protein after all phase 1, phase 2 trials, and you are producing large amount bulk the proteins like we have for hepatitis B virus, for example. And then you are injecting the protein in the body, and that is causing your protecting an antibody in the body, and you are now protected from the infection of the virus. So the two companies, one in China and called this Sinovac. Okay. Now we have another kind of vaccine that is called attenuated, and that is the front runner in our India. That in the collaboration with ICMR and NIV Pune, and Bharat Biotech is now going to produce, or the production I think has already been started. I'm not very much aware of the current scenario. So Bharat Biotech is now producing the vaccine. So what is attenuated vaccine? So attenuated vaccine is, you have, instead of the genetic material of the virus, instead of a fragment or a small piece of the immunogenic protein of the virus, you have the whole virus. You take the whole body, right? And you attenuate it. Attenuate, you either you inactivate the virus, actually. You heat kill the virus, or you treat the virus with some chemical, and actually it is a dead body being used as a virus. Another strategy is you make some genetic changes in the viral genome, so that if the virus is produced in the culture system inside the laboratory, the virus is not infectious. It is live, but it is not infectious. So that is called live attenuated virus, and this is called dead attenuated virus. So that Bharat Biotech we have now in India. And another one is repurposed vaccine. And repurposed means the drug or the vaccine which is already approved for another disease, working very well, very effective, and you are, based on your studies, you find, okay, let us check this one for a new disease, and that is also working. That is called broad spectrum drug, or multifunctional drug, cross disease drug or vaccine that is also going to act on another disease. So we have that BCG vaccine. So BCG vaccine is also acting very good in COVID-19 patient, and in research it has been found that the children or the population who has been already vaccinated in childhood or in adulthood, whatever, with the BCG vaccine, they are less susceptible to COVID-19 disease. So now, this is the current updates on the vaccines, what is going on globally. Okay, so now I think come to my summary points to conclude. So what we have seen, that SARS coronavirus 2, it has a potential zoonosis in bat, and there are many subtle genetic changes in the genome, especially unique mutations in surface 3A, 3B, and insertion of PP1B in other ORFs, operating frames. And these are the genetic basis of high transmission and high infectivity of this virus, and also the aggressiveness of this virus in terms of disease severity. And it has also passed to immune system evasion or inhibition by immune system, and it is getting very easily adapted to the human body, and also causing broad tissue tropism or cross tissue tropism, and leading to multi-organ pathogenesis in the infected person. And hundreds and hundreds of drugs, including vaccine candidates, are registered in different countries. And I told you in previous slides, some are in preclinical trials, some are in phase 1, some are in phase 2, and in the immediate last slide, I showed you the most leading vaccine candidates. We are very soon coming in the market, and the drugs I told you, remdesivir or favipiravir, that has been already approved for emergency use in USA and Russia, and the phase 3 trials, Moderna, Oxford, and Pfizer vaccines, which have shown about 94 to 95% protection. But of course, we have some little data controversy recently regarding the Oxford, and at some places the trials were stopped. But I think it is now resolved. And lastly, until we get the vaccine in the market, I think based on the news and the data, we are not going to get any vaccines by March or April next year, 2021. And even if we get the vaccines by February, March next year, the common people are not going to have access of the vaccine at least by the mid of the next year, by June, July, or something, right? So what we should do now by the time we get vaccine? So that is the a childish question. We have to have recommendations of MSP, not to confuse with the currently going on the agitations and the farmers things MSP regarding the agriculture. So I have made it like MSP for us, for researchers, for us, for scientists like us to recommend is, use mask. Face mask, that is the most important and most strongest protection device we have, mask. And then you have sanitization. Use sanitizers, not only for your hands, for all your things you touch anywhere, inside house, outdoor, in your office, in your university, classroom, everywhere. And the last one is P. So I say it physical distancing, because it is not in a real sense social distancing. Social distancing means now what we are talking to each other from miles, thousands of miles apart, and there are many people who are joining us from home. Some are present in maybe in the auditorium, in the hall, there in your place. So we are socially connected. We are not socially distant. We are actually we need to say like physically distance, physical contact, not social contact. So the right term is physical distancing. Okay, fine. So now the recent updates I should also include here. So recently, as for December 2nd news, UK has authorized Pfizer's vaccine for emergency use. And then SK Biosciences has entered on phase 1 clinical trial. This is on as per December 2nd. And this is the old news regarding Cuba announces 2 phase clinical trials on 30th of November. And Moderna request emergency use authorization, but so far it has not been given. In comparison, Pfizer has been given authorization by Britain, by UK, 2 days before. Okay, so I think I have concluded my talk, and now we should have some discussions if your time permits. And finally, I say thank you very much, with my prayers for my family, your family, everyone on the planet. And I should say thank you very much. And I should quote 2 very important mantras and very important, prayers, dua. One from our Sanatan, our Vedic text, and one from Quran. Meaning is, oh God, oh Ishwar, oh Allah, lead us from ignorance to earth, and lead us from the darkness to light, and show us good path, and get us very soon out of this global pandemic crisis. Thank you very much.
Thank you, sir. Thank you very much for updating our knowledge on the COVID-19 and its related informations. If there is any query, the same may be asked right now. Our speaker is with us.
Yeah.
Any query, please?
I'm there.
Apart from this, if further any query is persisting, this may be addressed over the mail that is event_sbt@iftmuniversity.ac.in. And now I would like to request Dr. Tanjil Ahmad sir for proposing vote of thanks.
Thank you, Professor
Please,
Mishra.
sir.
Thank you, Professor Mishra. The current circumstances we live in bring a lot of uncertainties and new challenges due to COVID-19. There is a need to improve knowledge for this pandemic disease. We have to take precautions and follow the safety guidelines to combat this pandemic disease. With this word, I would like to extend a warm good evening to all of you. Honorable Chancellor Sir, Shri Rajiv Kotiwal, Pro-Chancellor Sir, Shri Abhinav Kotiwal, Vice-Chancellor Sir, Professor M.P. Pandey, Pro-Vice-Chancellor Sir, Professor Rahul Mishra, Registrar Sir, Shri Sanjeev Agarwal, Directors of different schools, faculty members, and dear students. On behalf of the School of Biotechnology, IFTM University, myself, Dr. Tanzil Ahmad, extend a heartfelt thanks to the key speaker, Professor Khalid Parvez. Thank you, sir, for delivering an outstanding content on COVID-19 and coronavirus, its pathogenesis and containment. I believe that this lecture was beneficial for all of us. Thank you, sir, for connecting virtually with us during this pandemic in a very short notice. I would like to extend my senior
Thank you very much. Yeah.
to Honorable
Thank you
Chancellor,
all of you and your
sir.
senior faculty on the screen. Dhanyavad. Thank you, sir, and giving me this opportunity to connect to my Indian fraternity, my own people, my own land.
May I ask
Yes,
the
sir.
mobile number of Dr. Khalid Parvez? May I take
Yes,
your
you,
WhatsApp number?
Yes, sir.
For
pleasure.
some correspondence regarding some webinar.
Yes, sir.
So
Yes,
from
sir.
where I can get?
Dr. Tanzil Ahmed has my contact numbers because he is my very good friend, you know.
Yeah.
So please, Dr. Tanzil. Yeah. So among your faculty, whosoever professors
Okay,
and doctors wants my number,
I have put in my chat box, so please give it to me, please. This WhatsApp number.
I will give it to them. I will give it to them.
Yes, sir.
Okay.
Yeah, my email ID, my WhatsApp, please. Yeah.
Okay.
It's my pleasure.
Thank you.
Thank you all from the.
Thank you. Namaskar.
Thank you all.
Yes, sir.
One more thing that I would like to share to the participants, that filling of the feedback form is mandatory. So each and every member, each and every delegate is requested to present the feedback form. We are sending the feedback form through the mail, and the participant can download their certificates from the mail. Thank you very much. And now I am on behalf of IFTM University, I am ending this. Thank you all.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you.
Okay, thank you.